Right, good afternoon, everyone. So today I'm going to talk to you a little bit about a new way of separating really small nanoscale objects, both based on their size and on their shapes. So prion-based diseases, like mad cow disease, chronic wasting disease, these things can be a huge drain on the economy. So for instance, in the UK, they lost about $4 billion trying to curb the spread of mad cow disease back in the 1990s. And today in Wisconsin, we're suffering from shouldn't get too enthusiastic. We're suffering from a similar threat from chronic wasting disease, or CWD. And CWD has been infecting our deer herds out here and has been a persistent drag on the hunting sector, for instance. And part of the problem is that there's no effective way to identify the disease other than simply inspecting brain tissue. So what are prions? Prions are a type of protein. When prions undergo a mutation or a change in shape, they become diseased. And this sets off a chain reaction. These diseased prions create more neighboring diseased prions, and the result is neurological damage, and then months to maybe a year later, you have death, right? So currently, there's no reliable technology for identifying a normal prion from a diseased prion. Now, one way to possibly do this would be if you could separate the normal prions from the diseased prion, and then maybe use some kind of antibody-based mechanism for identifying them, right? But the difficulty is that both of these prions are really small. They're roughly the same size. It's only the shape that changes. And there's no real technology for separating small nanoscale objects that are similarly sized but have different shapes. So what we do have is one of the most commonly used techniques for separating objects at the micro and nanoscale are what are known as membrane filters. So a membrane filter works really well if you're trying to separate roughly spherical shaped objects that are about a micrometer or larger in size. Things get harder when you are dealing with objects that are a lot smaller than a micrometer. And prions are tiny. They're about 1/100th of a micrometer in size. And not only are they small, the other difficulty is it's a little easier to separate objects by size. It's a lot harder to separate them by shape. Right? So a lot of currently existing membrane filters have either randomly shaped pores, or you have precision filters with circular or square pore openings. But none of these technologies are good at separating objects by shape. So here's a picture of a traditional precision opening membrane filter. As you can see in this picture, the membrane's fairly thick. The pores are really small, narrow, and long. And because of this pore geometry, it takes a really long time to filter even small amounts of fluid through these long and narrow pores. The thicker the membrane and the narrower the pores, the longer this process takes. With a traditional filter, if you're trying to sort particles with pore sizes of 50 nanometers or smaller, this can take tens of minutes, even hours. So our technology is different, and we believe we can easily solve this first problem of flow rates. Okay? So what we have developed is an extremely thin membrane, a lot thinner than the conventional membranes, that are just as robust despite the small thickness. Furthermore, these extremely thin membranes that we make, which we call nanomembranes, have what we call angled pore sidewalls. And what this does is it significantly reduces the impedance against fluid flow. And so we can flow fluids much faster because of the reduced thickness and the angled sidewalls. So not only can we filter things more rapidly, Also turns out the technique that we use for creating the pores in these thin membranes allows us to change the shape of the pores, the opening shape. And therefore, that allows us to customize these membranes to sort different objects by their shape in addition to their size. So for instance, we can make submicron square openings, like the picture on the left indicates, with really high tolerances, few nanometer tolerances on the size of these openings. But we can also, for instance, make rectangular slots. And if you look at the picture up top, it shows you that these pores have these angled sidewalls. So you have a larger opening up top and a smaller opening at the bottom. And we have really precise control over the length and the width of these slots. And that therefore allows us to separate, for instance, what we call low aspect ratio objects, things that are more spherical, from higher aspect ratio objects, things that are maybe elliptical or flatter. We can take this technology a step further and make more complex shapes. Right now, what we're working on is making more intricate and complex shapes. We're working on, for instance, this picture is at the micro scale, but we're pushing this down to the nanoscale regime. And where we're going with the current membranes we're working on is primarily to target the separation of microbes. So for instance, microbes like E. coli and Salmonella, these are rod-shaped bacteria, whereas something like a Staphylococcus is a more globular bacterium. And so by creating different kinds of membrane filters, we can separate these different kinds of pathogens from each other. The hope is that we can push this technology further, make much smaller pores, and then separate much smaller biological species like prions and other kinds of proteins. So our filters can easily be incorporated into a traditional filtration setup. And because of their low flow impedance, what this means is we can introduce our filters in what we call stacks without significantly reducing flow rates. And in these stacks, you can have filters with different pore opening sizes and shapes and simultaneously separate, trap different kinds of pathogens based on their size and shape. So we currently have funding to develop a water testing kit, a portable water testing kit that allows you to go out into the field and have a portable system to simultaneously separate and identify different kinds of pathogens. I'm going to skip through the market size. Now we'll do this. Right. So what I've been talking about so far is a technology that lies sort of on the cusp between a number of different biological separation markets that are all multi-billion-dollar markets. However, we see this as a platform technology. We see not just biological applications, other ones including cancer biomarkers or pore-based DNA sequencing, but also completely or inorganic systems. In particular, we look at microelectronic fabrication, and we are exploring, for instance, printing of aligned carbon nanotubes in microelectronic circuits. Our team also includes Mark Levenstein, who is at the back over here from the biology department at UW-Platteville, and Paul Evans, who will be joining us shortly from material science and engineering here in Madison. And we've also had several really good dedicated students from UW-Platteville, all undergraduate students who've been working on this project. But to take things to the next level, we realize we're going to need postdocs and graduate students, and therefore we're looking for funding on the order of about $600,000 to hire additional researchers. And we're also hoping to explore other technologies with further researchers and companies. All right, I will stop here. Thank you.
So we have time for one question. Does anybody have a, don't go anywhere. Do we have a question? So one of the things that, so I have a question, or maybe more, if you could just expand on your lab. I think it's amazing what you do at your lab with undergraduate support. So how long have you been working on this area, and what type of funding?
In this area broadly, I would say for the last 7 or 8 years. It started off as more fundamental investigations of nanomembranes. But then we've been, over the last 2 or 3 years, working on some of these applications. And the funding has been largely from WISIS, and then more recently from UW System through a Regent Scholar Grant.